Welcome to this MOOC on electron microscopy. The course is split in 3 parts, which will be presented successively by myself, Alain Brisson, by Veronika Kralj-Iglic, and by Ishi Talman. The outline of the course. First, a short introduction to electron microscopy, followed by the presentation of the two main techniques in EM, namely transmission electron microscopy and scanning electron microscopy. And for each technique, distinction between the conventional methods or room temperature methods and cryo methods. First, let's summarize the basic principles in electron microscopy. In an electron microscope, it is a scheme of a transmission electron microscope. So in an electron microscope, electrons are emitted by a heated filament. They are accelerated with a certain voltage, and they are focused on a sample. Electrons interact with the sample, and a magnified image is formed by means of electromagnetic lenses, and the image is recorded with a camera or a detector. It is very important to note that the electron microscope column is placed under high vacuum, and this point is critical for biological samples which are hydrated. Depending on the sample composition and thickness, electrons have different fates. Some electrons go through the sample without interacting with it. These are the unscattered electrons or transmitted electrons. Some electrons interact with the sample elastically, which means without exchanging energy with the sample. And some electrons interact with the sample inelastically, which means that there is a loss of energy upon interacting with the sample. The energy deposited in the sample is responsible for radiation damage or beam-induced damage. Inelastically scattered electrons give rise to various types of radiations, including secondary electrons and X-rays, which are used mainly in scanning electron microscopy. There are two types of electron microscopy techniques, to which correspond two types of electron microscopes: transmission electron microscopy, TEM, and scanning electron microscopy, SEM. TEM is used in the analysis of thin samples. Thin means below about 500 nanometer in the case of biological samples, which includes proteins, viruses, membrane fragments, and EVs. TEM applies also to thick samples like cells and tissues after their size is reduced, in general by embedding the sample in a resin and cutting thin sections. On the other hand, scanning electron microscopy applies to thick as well as to thin samples without size limitation. This includes tissues, cells, and EVs. A large variety of preparation methods are available for imaging biological samples in EM. Two main families of methods are distinguished: conventional methods, or ambient temperature, room temperature methods, which apply to air-dried samples, and low temperature methods, or cryo methods, in which samples are prepared and observed in their hydrated state. After this general introduction on EM, I will present the application of transmission electron microscopy to the analysis of EV samples. Two parts: conventional TEM, negative staining, and cryo-TEM. Negative staining is the most widely used method in TEM at room temperature. Its principle is simple. An EV sample is mixed with a solution containing a negative stain. A negative stain is a salt which contains one or several heavy atoms. The main negative stains are listed here: uranyl acetate, uranyl formate, sodium phosphotungstate, and some others. Negative stain ions distribute over the hydrophilic surfaces and cavities present in the sample, and upon drying, negative stain will form small clusters of about 1 nanometer size, which deposit locally. These negative stain clusters, which contain heavy atoms, scatter electrons more than the surrounding unstained areas, which provides image contrast. In practice, let's deposit small volume, 2 to 10 microliters of sample, on a carbon-coated EM grid. For a few minutes, let EVs and other components adsorb on the carbon film. Remove the excess liquid with a filter paper, and before drying, add a small droplet of the negative stain solution and incubate for a few more minutes. Remove the excess liquid with a filter paper, dry the material deposited on the EM grid in air, and then the grid is introduced and observed in the microscope, or it is stored at room temperature and observed later. This is one negative stain protocol. There are many variants. What are the advantages of negative staining in EV analysis? The sample volume required is small, just few microliters. The sample preparation time is short, few minutes. In fact, the main advantage is that EVs are imaged as single objects, and therefore TEM provides information on the size of EVs, their size distribution, with a resolution of about 2 nanometer, which is limited by the size of the heavy atom clusters. Provides information on EV morphology and on the sample heterogeneity to a certain extent. Let's look at some typical examples of negatively stained EV samples. On the left, an image from EVs from ascites of a melanoma patient, and on the right, EVs from human urine. On these images, EVs appear as roundish objects with a size ranging from 70 to 200, 300 nanometers, which is quite typical for EVs. And as you can see, many EVs present a characteristic cup-shaped morphology. This corresponds to the collapse of spherical vesicles, which occurs upon air drying. What are the limitations of negative staining in EV analysis? The main limitation is that EVs are imaged in a dry state, and drying is responsible for some structural damage. Drying alters EV morphology. The cup-shaped morphology that we just described is indeed an artifact caused by drying. Drying is also responsible for some shrinkage of EVs. EVs cannot be distinguished from non-vesicular particles, and EVs may form aggregates due to the artifactual concentration of EVs occurring during liquid evaporation. In addition, the distribution of EVs over EM grids is often heterogeneous, due to the heterogeneity in the adsorption properties of carbon films. And very important, TEM does not provide the absolute EV concentration. Altogether, EV is a low throughput technique in the sense that information is in general based on a small number of individual EVs, say less than 100 or few hundreds. Let's turn to Immuno-gold TEM. The objective of Immuno-gold TEM is to identify the cellular origin of EVs or their membrane compartment of origin, in general, in a sample containing different EV populations. The principle of Immuno-gold TEM is similar to immunofluorescence microscopy. There are two labeling tools: antibodies, recognizing a specific receptor or antigen exposed on the EV membrane surface, which are used to identify a specific EV population, and gold particles of 5 to 20 nanometer in diameter, which are conjugated to an antibody or a protein. Just because gold is easily detected by electron microscopy. In practice, there are two alternative approaches. Either a one-step immuno-gold labeling approach, in which the sample is mixed with a primary antibody which has been directly conjugated to a gold particle. This approach is rarely used in conventional TEM negative staining. The second approach, which is almost exclusively used at room temperature TEM, involves a two-step process. In step 1, an EV sample is mixed with a primary antibody. The excess primary antibody is eliminated after incubation. In step 2, a secondary antibody conjugated to a gold particle is added. The excess secondary antibody may be eliminated after incubation, although this is not strictly necessary. And finally, the sample is negatively stained. Here are representative images of immuno-gold labeling of EV samples. On the left, EVs from FCF7 cells labeled with an anti-CD63 antibody. And on the right, EVs from activated platelets labeled with annexin-5. By the way, annexin-5 is not an antibody, but a protein that binds to phosphatidylserine lipids, which is therefore used, and annexin-5 is therefore used to identify EVs exposing phosphatidylserine. As you can see, in both examples, labeling is highly specific, and in addition, the majority of these EVs are labeled, indicating that the isolation procedure was successful. This slide summarizes the advantages and limitations of immuno-gold labeling TEM. Immuno-gold labeling allows to identify or phenotype EV populations, to characterize EV populations in size distribution, morphology, and determine the relative number between immuno-labeled EVs and unlabeled EVs. The limitations are the same as discussed before for negative staining. Let's now switch to cryo-TEM applied to the analysis of EV samples. Principle of cryo-TEM. A thin film of sample is quickly frozen by plunging in a cryogen. This induces the transformation of liquid water into solid ice, also called amorphous or vitreous ice. And EVs and the other components contained within the sample are frozen and maintained in their native hydrated state. In practice, a small droplet of sample, say a few microliters, is deposited on an EM grid generally coated with a perforated carbon film. The grid is plunged into liquid ethane, cooled down by liquid nitrogen at a temperature below -170 Celsius degree. Then the grid is stored under liquid nitrogen until its observation in a cryo-electron microscope, where it is also kept at low temperature. It is thus critical that after freezing, the grid is always kept at low temperature at every step. What are the advantages of cryo-TEM in general? Cryo-TEM is the least invasive EM technique. There is no drying, no staining, no chemical fixation. Objects are observed in their near native hydrated state, and the contrast of cryo-TEM images reflects the intrinsic composition of the specimen. Now, the advantages of cryo-TEM in EV analysis, there are many. First, cryo-TEM resolved the lipid bilayer, which is the hallmark of lipid vesicles and EVs. This is very important as it allows to distinguish EVs from non-vesicular particles like lipoproteins. Let's look at some examples already. On the left, an EV from pure human plasma, and on the right, a group of EVs from a glioma stem cell conditioned medium. On these images, all EVs present two dark lines separated by about 4 nanometers at their periphery. This constitutes the absolute proof that these particles are surrounded by a lipid membrane. The resolution of these images is about 1 to 2 nanometers. Next example. This example illustrates the fact that cryo-EM allows to distinguish true EVs from non-vesicular particles. Here there are two particles of about the same size, 200 nanometers, from human plasma. The particle on the bottom is surrounded by a lipid bilayer. This is therefore a true EV. The particle on the top is not surrounded by a lipid bilayer. Therefore, this is not an EV. By the way, this is a lipoprotein complex. I insist here, cryo-EM is the only technique allowing such a distinction. Another important advantage of cryo-TEM is to provide reliable information on the EV size and on the EV morphology, as illustrated here. On the left, the size distribution of EVs from activated platelets using different activators. And on the right, an EV with a tubular morphology, several micrometer long, as you can see here, in fresh human plasma. Another advantage of cryo-TEM is to allow characterizing the heterogeneity, the homogeneity or heterogeneity of EV samples. Here, the bottom, the presence of EVs with multi-layered membrane structure, and on the right, the presence of aggregates. These objects are more or less frequent depending on the treatment used during EV isolation. A major advantage of cryo-TEM is to enable imaging EVs in complex media, like pure body fluids, as we've just seen, in the presence of high protein concentration, avoiding EV isolation methods like ultracentrifugation, size extrusion chromatography, or filtration, methods which are prone to induce various types of structural damage to EVs. What are the limitations of cryo-TEM in EV analysis? EVs are soft objects which are sensitive to mechanical stress, and therefore EVs may flatten down during the thinning of the liquid film just before freezing. This is more likely to affect the large EVs. Frozen hydrated specimens are highly sensitive to electron beam irradiation. Yet, this is not a real issue, as cryo-electron microscopes are equipped with systems allowing image recording under minimal electron dose. More importantly, cryo-TEM is a low throughput technique. Only few samples can be analyzed per day. This is partly solved with the last generation electron microscopes, which are equipped with automated grid exchange systems. Still, cryo-TEM remains a low throughput technique. Let's turn to immuno-gold labeling in cryo-TEM. In this case, it is much preferred to use a one-step labeling approach in which the primary antibody or marker protein is directly conjugated to a gold particle. In doing so, avoids treatment required to eliminate the excess of primary antibody, like ultracentrifugation or filtration, which are lengthy and may induce structural modification. On the right, this image from human plasma shows two EVs. The small one on the top is densely labeled with gold particles conjugated to annexin 5, as we saw before. So this means that this vesicle, this EV, is phosphatidylserine positive, while this vesicle in the bottom is not labeled, not a single gold particle. Therefore, it does not expose phosphatidylserine. Here is another example of a double immuno-gold labeling study applied to EVs derived from human platelets. Two types of gold particles, large gold particles of 10 nanometer diameter, which are conjugated to an antibody against the antigen CD41, which is a specific antigen specific of blood platelets. The small gold particles, 4 nanometer diameters, are conjugated to annexin 5, therefore labeling phosphatidylserine EVs. And this image provides a clear demonstration that the three EVs are both from platelet origin and expose phosphatidylserine. The last example here, to make a comparison between immuno-gold labeling in conventional methods, negative staining, and cryo-TEM. If the same sample, human plasma, was used, both EVs are densely labeled with annexin 5 gold particles. But on the left, the EV is extensively collapsed so that the amount of stain deposit is highly variable over the EV, which makes the image interpretation difficult, although this is an excellent image, I must say. On the right, the spherical morphology is ideally preserved, which simply makes the image interpretation much simpler. Advantages and limitations of Immuno-gold cryo-TEM, but basically these are the same advantages and limitations as discussed before for Immuno-gold TEM negative staining. But the conjugation of antibodies or proteins to gold particles requires some specific expertise. Well, now we will move to the presentations of scanning electron microscopy technique, which will be handled first by Veronika Kralj-Iglic and second by Alain for cryo-SEM.
Scanning electron microscopes use electron beam to image samples. Electrons interact with the sample and generate signals in the form of secondary electrons, backscattered electrons, and X-rays. Secondary electrons originate from the surface, that is, few nanometers deep. They are a result of inelastic scattering and have lower energy than incident and backscattered electrons. They originate from atoms of the sample and can provide the details. Backscattered electrons originate from deeper regions, that is, few micrometers deep. They are reflected back after elastic interactions with the atoms in the sample. They are sensitive to differences in atomic number. The higher atomic number, the higher is the signal. Deceleration of electrons upon interaction with the sample is a source of continuous electromagnetic wave spectrum called Bremsstrahlung within the X-ray wavelengths, which can give information of elemental constitution of the sample. The size range of the X-ray and electron wavelengths enables observation of extracellular vesicles, which are too small to be observed by visible light microscopy. However, to reach the sample, electron beam must not be disturbed and therefore should travel in vacuum. Extracellular vesicles are formed in aqueous media, which must be removed. To conserve the shape of the vesicles and to improve the quality of the image, the samples are elaborated with fixatives and stains. Here we briefly show how the sample is fixed with osmium tetroxide on a paper filter. Osmium tetroxide is toxic, therefore the procedure is performed in a hood. Nitrile gloves are used. Yellow arrow points to a chamber with a hole on top through which the compounds can be added by the syringe. The chamber can be disassembled to place the filter. The sample containing vesicles is placed into the chamber to cover the filter paper. Osmium tetroxide solution is added on top and left to react with the membranes for 2 hours. Then the solution is pushed through the filter by syringe, hopefully leaving fixed vesicles on the filter. The chamber is disassembled and filter is subjected to consecutive bathing in graded solutions to remove water. Filter spends about 10 minutes in each solution. Then it is left to dry in air overnight. The dried sample is mounted to a metallic stand and before being inserted into the observation chamber of the scanning electron microscope, it can be sputtered by a heavy metal. It can be seen that the procedure is lengthy and needs a skilled operator. There are several steps in the procedure where sensitive vesicles can be damaged, destroyed, or lost. First images of the plasma isolates from our laboratory were made more than 10 years ago in Åbo Academy University by Vit Schuster, then a PhD student, under guidance of Henry Högerstrand and his team. To observe first images of plasma vesicles in Ljubljana, we needed about a year. However, the result of scanning electron microscopy imaging is worth every effort. The resolution is sufficient to observe individual vesicles. The images give information on three-dimensional shape of vesicles. Although there may be some shrinking of the samples during drying, the size can be estimated. Images may capture hundreds of vesicles so that homogeneity of the sample can be observed and cells can be captured in different phases so that we can observe the budding process. We have observed isolates from cells treated by staurosporin to induce apoptosis. Imaging of erythrocytes treated by detergents revealed how buds are formed on the top of echinocyte spicules. We have imaged these vesicles directly in the tube in which they were isolated by cutting it. Elongated and oriented vesicles indicated that they were formed due to shear stress during the centrifugation. The same shapes of vesicles were found in isolates from different sources, and these shapes agree also with theoretically calculated shapes of minimal membrane energy. The invaginations are not artifacts of preparation, but the intrinsic equilibrium shapes of vesicles subjected to different properties of the membrane and of the surrounding solution. The limitations of the method are mainly in demanding preparation, which can damage or lose vesicles. Also, fragile internal structures cannot be revealed by classical scanning electron microscopy.